fellow and all organizers. This has been a really exciting interdisciplinary experience so far. So a lot of conversations that I don't always, engage in. Can you hear me okay even with the mask? I might also take it off. Okay. So what I'll talk to you about today is work that we're doing in my lab to try to build links between the brain and behavior. So how can we interpret what populations of neurons or what neural activity in the brain is actually related to, and actually use that to build inferences about what the organism is doing. And so the sort of motivation of this work, as I said, is how do we take recordings from the brain and try to use that to make sense of what the brain is doing. And in many cases, we do this through this mode of neural decoding. We actually heard a little bit about this in Maria's talk, this idea that I can take activity from populations of neurons in the brain and then try to build a model that I can use to relate that to some downstream behavior. And so one of the major challenges in doing this within neuroscience stems from the fact that neural responses, so what we actually measure in the brain, can be highly variable. So even when behavior can be quite stable, if we go in and measure the responses of individual neurons, their spiking activity can look quite different. So the observations that we're getting seem to be such that there's no way that we could actually, make a reliable link between these activity patterns and some underlying stimulus that we want to decode about the environment. And this is true even when we use very, very simplified stimuli. So, for instance, up on the right-hand corner, I'm showing an example where, to visual neurons, maybe we just display a grading pattern of a particular orientation or a particular frequency. And you would think, okay, well, this is a very simple stimulus that we're presenting to the brain, so probably the neurons should kind of have the same sorts of responses, but indeed we don't find that to be true. And of course, building those stable masks between the brain and behavior becomes even more challenging when we have more complex stimuli. So instead of just gradings, if we're talking about naturalistic behaviors that an animal can instantiate, this of course is even harder to try to learn this mapping. And so one of the, really exciting observations and something that has, at least given us some hope that this could be possible is that if instead of, sorry about the volume control, so if instead of considering a single neuron at a time, monitoring its spiking activity and then trying to relate that to some stimulus, instead what we found is that if we observe the large populations of neurons instead of single neurons, and then look at the sort of collective dynamics or responses that they all have, it actually looks a lot more stable. So this picture that I'm showing here is just now if we look at this population level, look at all the kind of joint dynamics between them, and visualize that as some sort of low-dimensional manifold or this latent space that all of the joint activities occupy, we actually see that the same stimulus produces very similar trajectories on these underlying latent manifolds. So what this means is that by sort of moving to this population level representation of neural activity, we can see this more stable and predictable map. But of course, when we're going into the brain, we can't measure every single neuron, and there's still a lot of changes that are happening in the brain and its activity that we think about as representational drift. And so despite the fact that looking at populations of neurons gives us a better handle on this mapping, we still have a lot of challenges in trying to put this all together due to which neurons we're actually sampling and how maybe those underlying representations could be changing over time. And so the approach that we've taken in our lab and in a number of projects where we're interested in kind of forming this relationship is twofold. So on one hand, we want to build new algorithms and new machine learning frameworks that allow us to get more stable representations where we give the same stimulus and we want to decode the same behavior and we have a stable latent representation, right? And so we're thinking of new ways to go beyond existing ML strategies or the ways that they're used within neuroscience and see what that can actually give us in terms of forming these more stable representations. And then at the same time, we're also in the lab interested in, once we have learned representations, how can we go about, actually comparing the distributions and in many cases aligning them or using that as a way to do diagnostics or understand when the brain has departed from its healthy state by looking at these comparisons of these underlying representations of brain activity. So what I'll talk about today is focusing just on the first aim. So how do we build new representation learning strategies that allow us to build this generalizable link? And so, as I said, already motivated, we have a number of challenges that, have made progress on this front challenging for the field. And, on one hand, as I said, neural activities could be shifting over time. And at the same time, we have this desire to build unsupervised representations that aren't necessarily biased by labels or specific human-driven inputs that we might think that the brain is trying to, encode within its neural representations. So this really has motivated the use of different unsupervised learning approaches for building representations of neural population activity. And so in the conference thus far, we've heard a number of talks that have, kind of discussed this generative modeling approach. And there's a number of different classes of models, whether it be autoencoders or variational versions or also generative adversarial networks. And all of these can be classified as a sort of generative approach for unsupervised learning. And in the context of neural activity, I guess I can't see the pointer here, but so in the case of neural activity, what an autoencoder approach might look like is if we start over here on the right, I'm showing multiple neurons that have been recorded over time, and we're going to essentially treat the observations of our neural population. Say there's D neurons, we can treat that as some sort of D-dimensional vector or a state vector that we're trying to distill and kind of embed in this lower dimensional space. And what our autoencoders and GANs are trying to do, they're trying to build a representation that allows you to produce realistic outputs that match the overall statistics of the input or the training data, right? And so reconstruction-based approaches will basically, plug your data in, go to some lower dimensional space, but then the goal in the end is just to be able to recapitulate back what our inputs were, reconstruct them under some sort of loss term. And these sorts of ideas of using even simple models like principal components analysis, and then going to, far more sophisticated deep learning versions of this, have actually given us a lot of insights already into, as I said before, the fact that, populations of neurons are actually encoding different information about their inputs, and they're doing it in this collective way, in a way that we can't actually see from individual neurons. So already using these sorts of approaches has given us a lot of powerful insights into the brain and neural circuits. However, often when we use these reconstruction-based objectives, we can tend to overfit to noise in our original training data. So there's been a lot of work that's been done in just the machine learning community to show that generative models often don't generalize to slightly different conditions. And part of this can be, thought about by the fact that, like if we measure the mean squared error, we're trying to minimize variance, that means that those sources of high variability, say it's just noise or artifacts in our data, they'll want to be captured through this model, right? And there's nothing to sort of de-incentivize the model from picking up on those fluctuations that are very specific to our original data set. So as a result, generative modeling approaches can fail to generalize, and in particular, the representations that they learn might not be well applied to new test data that we haven't seen at training time. So that leads us to a new class of unsupervised learning methods that are we talked about them yesterday. We discussed the idea of masked language models. These are part of classes of self-supervised learning algorithms that take a slightly different approach, but still try to learn representations from our data without this kind of reconstruction-based objective as its main goal. And so what do these self-supervised learning approaches actually do? So one of the core sort of mechanisms that allows these models to build rich representations is the use of what are called augmentations, or essentially taking classes of transformations that we apply to our input data to create a new view of the original data that we started with. And so some examples are shown over here on the right-hand side, where we could start with, a picture of a cat, and then we could apply two different transformations of that cat out of a set of known transformations that we have in hand. So in this example, I've created two different views of our original image. And in the first case, if I apply transformation T, maybe I just change the colors within the original image, right? So I'm creating a color-based augmentation. And then in the other case, T prime, I've applied an augmentation or transformation where I basically randomly go to a different pixel in space, zoom in, and then crop. So what we can see is that, for these different types of transformations we've applied here, you'll create all these different instantiations of your data, but in all these cases, we still have preserved the cattiness of our original image. And so after we, apply all these different transformations to our data, we get augmentations. Now we're going to define our loss function, not in terms of reconstruction, but actually what we're going to do is we're going to try to find a representation that allows us to map all of these different augmented views to similar parts of our data space. And the sort of intuition behind this is that if we apply these transformations in such a way where we still preserve the cat, then what our algorithm has to do is find the common thing that is not changing amidst all of the other perturbations that we're applying. And so, the outcome of this is really trying to find some representation where you extract the common piece of information amongst these different transformations. And there's a number of different algorithms that are used to apply these same sorts of principles. And I'm just highlighting one called bootstrap your own link or bio over on the right, where the goal of this neural network, as it kind of said in words, is we're going to pass each view through our encoder or through our neural network parameterized by, well, here F, we get representations of each view, Y and Y prime, and then our goal is just to minimize the deviation between those augmentations. But then during training, we keep on constantly creating new augmentations and different transformations of our inputs. So each time that it sees these different views, it's actually getting, two completely different images that it has to find some sort of common information across. And I will get back to neuroscience. So these are just some examples of the extreme types of augmentations that are used within image recognition. So, if you're interested in doing object recognition and you're using something like ImageNet, the sorts of augmentations that are used can be very extreme in the sense that they can actually start generating data that doesn't match the natural statistics that we would expect to see at test time. So for instance, here, if we look at this cutout augmentation, none of our testing images will have a huge gray square in the middle, but during training, we can apply these, potentially very wild transformations of our data as long as they still preserve the same underlying semantics that we care about, or the information that we're trying to decode is sort of consistent and common throughout. And so these are examples up top of, augmentations that are used, for instance, in solving ImageNet. But then when we move to new domains that, either where it's not image recognition, or maybe we have different image statistics, like say in medical imaging, knowing the right augmentations and transformations to apply to build in invariances that you care about is not a trivial task, right? So we were excited about this idea of being able to build representations to this framework. And our first question was, okay, well, how can we apply these same principles within our original problem that I stated, which is, taking populations of neurons and trying to relate them to behaviors or stimuli or input. So now, when we're trying to do this in this new domain, what are the appropriate transformations that we need to apply? What are the right augmentations? And how can this actually give us a better representation of what neurons and neural circuits are doing? So to answer that question, we set out on a journey, at least initially kind of coming at it from a bit of a engineering perspective, right? So we can try different types of transformations that are, motivated from underlying neuroscientific principles, and then see whether or not they preserve the information that we care about within the neural population of interest. And so to start, we wanted to create sets of augmentations that make sense given what we know about neural coding, but are also quite simple and general and might actually apply in many different types of areas of the brain, or if we're decoding vision versus movement, we could imagine that these same transformations might apply. So much like what we heard about these masked language models that try to build representations by essentially kind of masking out different words in a sentence and trying to build a model that could predict or fill in those missing entries. So we can take a similar approach here, but now instead of words, we're actually talking about neurons and their firing patterns. And so our assumption is that, as I said before, we have a population of neurons, it's encoding some information, but it does it in a distributed and a redundant way. So the first thing we can do to build these views that I talked about of our data is actually just mask out different subsets of neurons from the population. And so what we're seeing in that case, when we pair, a brain state in one condition with some missing neurons and the same brain state with other neurons missing, is we're asking, okay, what is the information that this circuit has that's common despite, turning off or on some sets of neurons? So dropping out neurons from our original data, and then using this as a way of forming this self-supervised representation. So that's the first thing, is masking out neurons and using that as an augmentation. And then we can also test the assumption that as the brain is encoding different stimuli or inputs, it's not dramatically changing its representation at each instant, and there is maybe some sort of smooth evolution of those neural states. And therefore, we might also consider just temporal augmentations, where we could pair nearby points in time and try to find the thing that's common, despite that sort of jitter, that temporal jitter. So I guess, to answer this first question of what are good augmentations for neural data, we wanted to test these general classes of neuronal dropout and temporal prediction as well. And then how can we say that they're good augmentations for neural data? As I said, we kind of take a more engineering approach here. So going in and, with a number of these different algorithmic frameworks or ways of incorporating different augmentations, we can basically, plug these things in and then ask whether or not the information that we know is roughly being encoded within the neural circuit is being preserved. So in our first case that I'm showing here, it's kind of a simplified setup, but it's one that you can imagine would be important in brain-machine interfaces, where you're recording from neurons within the primary motor cortex of the monkey brain. And we know that this area is important for and controls downstream movements. And so we have animals that are performing a center out reaching task. They basically get a cue, and then they're going to make a reaching movement towards one of 8 different targets. And at the same time, we're recording from their brain. And now in the kind of supervised decoding case, we would take neural patterns when an animal is reaching towards one target and try to build a model that could, predict that target, right? So we'd have a supervised setup. And in this case, when we use the self-supervised approach, we're going to create these augmentations that I talked about, build this latent space that has this nice alignment property across the augmentations. And then what we'll do is that's completely unsupervised, right? We haven't used any labels. And then we're going to freeze our model. So I guess, as Matthew was saying, we'll lock up our model. We'll put a lock on it. And then we can plug our data through this frozen model. So it's already learned its representations. It gives us some embedding of our data, and then we append just a linear readout from that representation space to try to predict these 8 different targets. So the model didn't use the target directions when it was training, but we can validate to say whether or not the representations that it learned are high quality and allow us to separate these different behavioral variables. And so that's actually, how you can test to see whether or not your unsupervised algorithm allows you to predict on this downstream task. So in this case, I'm applying this algorithm or these ideas on 4 different data sets from 2 different animals, so Chewy and Mihi, and comparing with like a supervised decoder that uses the labels in this end-to-end manner, an autoencoder, so a generative model. We have a more sophisticated generative model, and then the 2 at the bottom, Sinclair and Bio, are 2 different ways of incorporating these augmentations through this representation learning strategy. So the first takeaway is that, on average, we actually find that these self-supervised methods tend to do almost as well, or in many cases better than a supervised decoder. So, going back to the title of my talk about how dropout can help us build more generalizable links between the brain and behavior, we can take away from this that on test data, through this unsupervised procedure, we've actually done a better job of being able to disentangle these different target variables, and on held out or test data, we can do a better job of generalizing, from this unsupervised representation. And then moreover, we found that dropout is already a very powerful mode or way to create augmentations of brain states that allow us to, pull out these different sources of information from these data sets. All right. How am I for time? That's okay. Or just tell me. 5 minutes? Okay. So I won't have time to go into the details of how we've sort of gone beyond these initial models that I described, but we kind of were motivated by this idea that, okay, we can create these augmentations, and if we know good transformations of our data, like for instance in the natural image case, we can zoom and crop and do all these things, then this can give us this kind of powerful representation. But in the case of these neural augmentations, they're a lot weaker, right? Like they don't have a lot of strong information already embedded in them. So we were interested in whether or not we could actually find other brain states that represent similar pieces of information at other points in time. So it's kind of this idea of going beyond just this local view that you might have temporally and trying to find other points in time that you could use as sort of instances for learning or prediction. And so just on the right here, it's like this idea that, okay, we started with this picture of a cat, we created an augmented cat, but what if we could find other pictures of cute kittens that look kind of similar? Could that help our learning procedure through these sorts of associations that might not be from the same exact instance. And so we developed a self-supervised learning algorithm called Meow, or Mind Your Own View, that tries to do this. If I can play it. I think it's like skipping over the videos. Sorry. It's not going for, oh, okay. Well, we could also just not look at this cool video that I made. I can still explain it, even though it's not going to graphics. But basically the idea is, you know, we have this algorithm or this architecture that allows us to solve this first problem of like these very local, just dropout based or you know, augmentation based learning, and then you know, we can even think about this as we're thinking about infants, just being able to predict from partial information what should happen next in your world is a relatively simple task, right? Like a very local way of building inferences. But then at some point, right, when we're able to, you know, have more memories or higher cognitive functions, we can now think about, like, when we see this cat, oh, I remember another instance where, you know, I've experienced a cat. And so we embed this into this algorithm by basically taking other examples from our data set. So we don't know the labels on them, like could be dogs or tigers or other cats, and essentially plugging those into our representation space that we've learned. And essentially, again, we have a way of just, you know, looking within the representation space, finding nearest neighbors in the representation space, and then using those nearby points in our representation as positive examples for these like more diverse views that aren't the same image, but could have similar semantics as learned by our algorithm. So essentially building in this kind of nearest neighbors based approach that can be plugged into this same framework. We first tested this in kind of more standard benchmarks where this has already been well established, showing that we could, you know, compete with a lot of the state-of-the-art methods at the time. And these are just some examples of, you know, the original samples on the top row, an augmented view of that same sample, and then the mind view that's selected by our algorithm. And then we find, you know, in the vision case, a lot of times the mind views are from the same class, and even if they aren't, they do have underlying semantic structure that's similar in both cases. So this is kind of a nice confirmation that the algorithm and this mining approach can actually give us something, something useful within visual representations. And so then when we add this new algorithm back into the picture that we saw before, we, you know, get across the board like improvements over vial, and then also in some cases where we weren't hitting the supervised baseline, we're able to then, you know, achieve those same overall accuracies in our simple reaching task. So in the next few minutes, I'll just kind of quickly tell you that we've started to use this in more complex and more naturalistic conditions, and also in other organisms, so in this case, rodent, both mouse and rat. And, you know, finding again that we can use these same simple augmentations and actually do a good job of decoding. But in this case, we, you know, we're recording from the rat V1 or the mouse hippocampus while the animal is just going about its business. So we're recording 12 hours. It's in a cage. It has objects in the cage. It can drink water. It can, you know, eat its pellets, etc. And but then when we're decoding here, I'm just decoding this very coarse behavioral label. So I'm just saying whether or not the animal is awake, in REM or non-REM. So we could say, all right, well, you know, that gives us some information about the quality of our representation, but clearly there's a whole lot more that could be going on under the hood that this decoding accuracy wouldn't tell us. And so we've started to dig into these latent representations and find that a lot of the clusters that are sort of revealed through this algorithm actually do coincide with relatively complex behaviors that we can see in the simultaneously recorded video from those same subjects. So here I'm just showing a few examples of some of the more complex behaviors that we've been able to pull out with this algorithm. And it's so preliminary work in the sense that we're going back and actually getting labels on all of those behaviors and seeing whether or not this algorithm is telling us something more, you know, and something informative about these really complex and naturalistic behaviors. Okay, so yeah, all kinds of cool stuff. And, you know, if we're talking about ethics and privacy and reading out brain states, yeah, I mean, this is it's like we can kind of, you know, we have a picture of a brain state, and we can go back to all of the other instances that we think are kind of related to that brain state, and then actually see the video corresponding to that. Okay. So this is still work in progress, but we think that this is a pretty exciting new way to use these sorts of dropout-based augmentations along with self-supervised learning as a way of building more robust pictures of neural activity. We've also combined this with generative modeling approaches to do disentanglement of different aspects of neural activity. And so here, just trying to separate the content from the style of movement representations in the brain. We talked a little bit about style in other contexts. Happy to talk more with those about this that are interesting. And then finally, I'll just end with some kind of open questions. So, you know, what I've talked about thus far is this idea of dropout as an augmentation has some underlying assumptions. And in general, this same principle could be used in many different ways. It's quite flexible, right? So you could think of an augmentation that you think, you know, may still preserve content or information within your data, and you could kind of potentially use this as like a virtual assay of how certain parts of information are being encoded within neural circuits. So for instance, our example of neuron dropout, we're sort of saying that, you know, the same piece of information is being encoded and is distributed across the population. And this gives us a way of actually testing that and testing the degree to which it's redundant or not. Okay, cool. So, and then finally, there are just some cool ideas floating around here, like could we actually come up with adversarial augmentations that might remove our ability to decode information, right? So kind of taking the other approach, not what can we do to preserve information, but what can we do to destroy it? And there's a lot of tools in optogenetics now that allow us like actual measurable ways of producing these perturbations and maybe testing the theories that come out of this. All right. So I'll just thank, you know, all the people that made this work happen. So this is a big collaborative effort with DeepMind and our collaborators at Washington St. Louis that collected all the data that I talked about in the second half of the talk. And then, of course, my awesome students over here on the left in the lab that, you know, really drove this work. And our funding. Thank you so much for your attention. Sorry I went over. We will take questions.
A couple times you talked about the populations of neurons. Could you explain what you mean by that?
Oh yeah. So all I'm saying, all I mean is just I've recorded from a number of neurons, and when I say population, it's always, you know, from one recording device that's kind of measuring locally in a vicinity. So the neurons that you're recording might or might not be from the same kind of underlying neural circuit, but given the levels of redundancy that usually when we record from, say, like 200 neurons in this case, they all are a part of kind of similar computations within that area of the brain. So yeah, population just meaning I have many neurons that I'm recording from simultaneously. So, oh yeah, so like all the data that I talked about in this talk is coming from like multi-electrode arrays that are implanted into different parts of the brain. So all the motor cortex, like they'll target a specific region within the motor area. And then you basically have an electrode array that can measure the potential of all of the sort of nearby neurons within that, within some small region around that implant. There's more, there are now approaches that allow you to actually measure from different regions of the brain simultaneously, but unfortunately, a lot of the electrical recordings are still kind of limited in how wide of a field of view, if you will, they can capture. But we're scaling it up as a field. Does that clarify? Or we can talk more.
It's really, really, really important. I'm really looking forward to talking more about neuroscience and questions. But I want to ask a question. So I really like the meow framework, which, if I understand, is neuroscience is augment the data set. Have you thought about using contrastive learning approaches where you have both kind of a close the nearest neighbor and something that's like farther away? And how should people this like farther away see? Would it matter how you feel it? Is it just like farther away?
Yeah, no, that's a great question. So just to kind of repeat that, the question was about how, or if we've looked at contrastive learning variants of this. And so the algorithms that I was comparing with, one of them is a contrastive learning algorithm called SimClear. And the difference is, you know, in the predictive framework that I highlighted here, we were only thinking about what are views that are deemed similar, right? Or these positive, I'm sorry, I'm like, I know you know, I'm just kind of giving that. But like in our framework, we were looking at things that we deem are similar, right? Or positive views. And then in the contrastive learning objective, in addition to having those positive views, you also have other samples from your data set that you just say are different, right? And one of the challenges with, you know, SimClear and with contrastive learning is often you have to have really large numbers of negative examples before you get one that's kind of that contributes to your learning signal significantly. So I guess just in practice, we found that these positive augmentation-based approaches you know, do outperform the contrastive learning. The question of, you know, why that's the case, or maybe our negative examples like aren't good enough, or are, you know, or too similar. So yeah, I haven't actually looked under the hood there, but at least when we're defining negative examples in that case, or the way that people do it in temporal or in time series learning is basically you have your small window that you define the positive view, and then almost anything, you know, outside of that small view is considered a negative example. And I think what we're showing with Meow is that there are a lot of things outside of that small window that actually are positive examples, and our algorithm is able to, you know, grab them and pull them closer. And but yeah, you could imagine using like a nearest neighbor approach, perhaps in a contrastive setup. And there are other works that try to improve upon just this random sampling by doing what's called hard negative mining. So essentially trying to find other examples that are close to the positive example, but are negatives. So yeah, I guess you could imagine different ways of doing nearest neighbor or other adaptive searches to find better negative examples, and then, you know, be able to also use that with this. Yeah, thank you.
So I have a question regarding, you said that neural world is like very complex and the patterns are not easily recognized. And the ways to find the pattern in the signals, right? By using the randomness of the signals, right? That's something. So the question is, are those patterns that that we subset, we see that they are the patterns that are defining particular action, right? So are those patterns always same for same action? Are we able to always find those patterns, right? And move the randomness to the sequence? Will it be always possible? And
I think I missed the end of it, but as I understood, you're asking, this question of I give you the same stimulus, I get some spikes, and how would it be possible to find like consistent patterns that arise from that?
So is it always possible to find a consistent pattern for a particular action? And those similar to the spike?
Right. Yeah, it's a good question. So, traditionally in a lot of systems neuroscience, the way that people go about, dealing with this variability is you basically will show exactly the same stimulus over and over, and then record the neural activity, and then you basically try to form some sort of mean or average to the patterns that are emitted over and over, right? And so, the picture that I showed, everything looks completely different, and so maybe it's just that you haven't had enough observations, but you keep on, applying the same stimulus over and over, and enough of those times you get something that kind of looks consistent, and so that when you do this trial averaging, you get a signal that's kind of indicative of the stimulus. And so in some cases that does work. There are other neurons that are more stochastic and where those trial averages are an even weaker descriptor of what that neuron does. But I think the idea that, I kind of have, and, the field to some extent has, is that because there is this redundancy within the circuit, let's just say a neuron is encoding, reach to the left, right? And in some of those instances, you see it firing and you see it, respond strongly when the animal goes to the left. But then there's other cases where a different neuron that's kind of encoding the same things might fire instead of that first neuron. And so if you just are measuring that neuron, it looks silent and it feels like, it's not actually representing this information any longer, but it's actually that there's this kind of maybe non-uniqueness in some sense about which neurons need to be activated, but collectively they all kind of are working together to produce the same sorts of outputs. So yeah, it's kind of hard to say, but yeah. Happy to talk more offline.
You intrigued me with your hint at adversarial stimuli, and I'm fascinated by those in the computer science context, but mostly to ask whether or not they exist in nature. Do you think that there are adversarial stimuli that are used as a strategy by, for example, pathogens, that you might actually be able to explain pathogenic states in the brain as adversarial stimuli?
Yeah, that's a really cool idea. And I haven't thought about it we haven't started to go in this direction in terms of designing adversaries. We have done a little work in just, instead of just having uniform dropout across the neurons, could you actually learn like a distribution of your dropout, where in the case where we want to preserve information, it's like, oh, these neurons kind of matter and I'll drop them out at this rate, and these other neurons don't matter as much, and so they'll be dropped out all the time, for instance. So you could imagine using, just in this simplified case, maybe an adversarial or just an information breaking prior to design those augmentations as well. So you'd basically know which neurons, if you drop them at all, will destroy your ability to decode. And I think this idea that pathogens, viruses, or even other sorts of disease that could happen over longer time scales, could have some mechanism to find, or could be viewed in some ways as adversarial augmentations. I think this also gives you some sense of what the neural circuit is already robust to, and then what it's not robust to. And maybe that could be helpful in designing, drugs in different cases, right?
Yeah, I mean, there's lots of adversaries that are neural adversaries, particularly for things that are altering behavior. For example, zombie ants with these fungal neural adversaries. So, yeah, how do they crack in and elicit a new set of behaviors? And
Yeah, I'd love to talk about that more. Cool, thanks.
Could you say more about, you mentioned the style. Can you explain more what you mean by style and how you're utilizing it?
Yeah, so it's kind of like a loose interpretation, but basically in this particular formulation, we're using the same idea of like the self-supervised loss that helps us get a good representation. And then basically we use a common and private encoder model to try to disentangle the thing that's learned through the augmentations from whatever other variants exist in our data. And so in the context of this sort of neural representation of movements, we can think about the content of it as which target are you trying to go to? So where, and then the style as being the actual dynamics of the reach and like how you get to that target variable. So we think about the dynamics as kind of being the style and this kind of finer scale, you know, information about that could be individual specific or even reach specific. And then the content is, you know, the actual reach direction. And then you have a way of kind of trying to, when we're learning the representation, disentangle these two different sources of information within two sets of variables within our data space.
Wonderful, thank you.
Thank you.
Wonderful, thank you.
Oh, sorry.